Well, hello everybody, and welcome to tonight's webinar, the Genetics for Reproduction, the Female Influence. My name is Andrew Byrne, and I'm a Technical Officer within the Southern Beef Technology Services Project, and I'll be facilitating tonight's webinar on behalf of the staff of Southern and Tropical Beef Technology Services. Tonight's webinar, the major presenter will be David Johnson. David will be familiar to many of you, but David is a senior scientist at the Animal Genetics and Breeding Unit, or ABRI, based here in Armidale. And tonight's webinar obviously focusing on genetics for reproduction and some overview of the work that they've done in Northern Australia. David has been involved in all three CRCs, and more particularly as a project leader for a lot of this northern reproduction and fertility work. So we're very fortunate to have someone of David's calibre with us tonight. We're very much looking forward to his presentation. Just before I hand over to David to get us underway, just give you a little bit of background behind our webinar course that is coming up. So tonight's webinar forms the first session in the Cutting Edge webinar course, which will be run over each Monday night over the next 6 weeks. And for those of you that are aware that the Beef CRC finished on June the 30th, so it closed its doors, and so we were very keen to ensure that a lot of the research outcomes in the last phase of that project were delivered to industry, and one of the extension exercises that we're doing under SBTS and TBTS will be this webinar course to make sure that any of the outcomes from the Beef CRC in the third phase are delivered to you and to the seed stock industry. For those that may not have joined us in this webinar course previously, the way this will work is you will be able to see David's presentation and hear from him as we go through. You won't actually be able to talk and ask questions, but from time to time we will break the presentation and take a number of questions. So you have the option of typing the questions in that you may have as we go along by using the screen in front of you. So there should be a screen on your computer where you can actually type in that question and press send. That will come through to us, and when we break the question, we'll ask as many questions as we can. One common problem with the questions which we find, just to quickly cover, is that question box often disappears throughout the talk. So if you click on the double arrows, that will come back up and you should be able to type the question in. So with that housekeeping out of the way, I'll just change the presenters over now to David so that he can get us underway with tonight. So we might have just a slight delay while we get this underway.
Back to you, Andrew. Are we up and running?
You're up and running, David. You should be right to go.
Yeah. Good evening, everybody. Thanks for joining this webinar. You'll probably be glad that you're not in Armidale. There's already a frost on the top of my car, so quite happy to be back in your own homes. I want to go through the genetics of female reproduction. I noticed a lot of new people on the webinar list, so I just want to go through some of the design so you understand some of the research we've done. It's specifically for results from the northern work, but I think the results are relevant for all breeds, and certainly it was done in tropical, but I think anyone with temperate breeds will also get a lot out of this research, and I think there's some take-home messages at the end that will apply to all breeds. So I just want to go through the genetics of reproduction from the CRC. Obviously this is work from a large number of people. I've been the joint program leader, but it's certainly involved a lot of people and also funding bodies, and we'll have just some acknowledgements at the end. In terms of what I want to do in this presentation, I want to go through the project. I want to talk about female recording, some key results. Obviously I don't have time to go through lots and lots of results, but just want to hone in on the female reproduction, look at the heritabilities, which is the degree of genetic control for some of these female traits, and some of the key trait relationships between the female traits, but also with some of the steer traits. And then I want to finish up by how do we start capturing this for seed stock and commercial producers? So what do you got to do as a producer to start seeing and capturing some of these results from the CRC? The opening here that led a lot of this research was selection for whole herd profitability. What are the genetic trade-offs? If we can improve steer performance, growth, feed efficiency, carcass, meat quality in the steer, what does that mean for female performance in terms of adaptation and growth, puberty, early reproduction, and lifetime reproduction? So if we select for steer traits, what happens to the cow traits, and vice versa? And that was the background for a lot of this work that we undertook in the CRC. So I just want to go through the design of the Beef CRC Northern Project. We worked with two tropical genotypes, the BRAHMAN and the tropical composite. The tropical composite was from the Northern Pastoral Group of companies, based in some cases around the Belmont Red, representing about 50% adapted genes. We used 50 sires per breed in a genetics design where we wanted to get 25 female progeny per sire. So we used industry and company sires. These were produced on the Northern Pastoral Group of companies. We used AI and natural mating, the AI to get genetic linkage across the stations. And we generated over 2,000 steers. All those steers were 120-day fed. We collected full growth, carcass, feed intake, and meat quality, MQ for meat quality. And the most important part was that we retained all females. And so the project had 2,200 females. At weaning, they were allocated across 4 locations in Queensland, and we kept those females for 6 annual breeding seasons until they were rising 9-year-old cows. Cows stayed in the project for their whole life. They were only culled if they failed to wean a calf in 2 consecutive years. So a very big study in terms of numbers from a genetics point of view, all pedigree recorded, and it's the heifer bit that I want to focus in on tonight. Just to give people a bit of perspective where this happened, the tropical composite breeding properties, we sourced those across Northern Territory through Queensland at these particular places. The Brahmans were bred at, across central Queensland places, and then at weaning we took each of the females and put them at 4 research stations: Toorak, Swans Lagoon, Belmont and Bryan pastures. So we've had across a range of production systems or environments, and we put Brahmans and composites at the different locations, noting that Brahmans were only at Swans Lagoon and Bryan pastures only had the composites. So the real strength in the design was the fact that we generated these families. So here's an example of Tartarus 3223 with his sons, his steers, and also his daughters. And so we had all these half-sib families where on average we had 20 to 25 steers, but we had 20 to 25 sisters, which is a very powerful design for looking at these genetics of reproduction and how they relate to the steer. Likewise, in the tropical composite, we had the Mount Eugene. Here's an example, 88145, his steers and his daughters. So obviously the bull, he has his genes in the steers producing the steer type traits, but likewise we've got the sisters out there doing reproduction in the tropical environments. So in terms of the genetic outcomes, we've got the steer type traits: growth, feed intake, carcass, yield, meat quality, and we've got estimating heritabilities and genetic relationships, and also overlaid was the gene markers. And then we've got the cow type traits: cow growth, body composition, puberty, adaptation and reproduction. Likewise, we wanted to see what was the heritabilities, genetic relationships between these traits, gene markers, but in terms of whole herd profitability, we wanted to estimate what is the genetic relationship between these cow traits and these steer traits. So can we improve one without affecting the other, and vice versa, because that's what we are trying to always do under our production systems. In terms of the steer traits, I could give a 3-day seminar series on those results. I just want to highlight here that we measured lots of traits and estimated heritabilities, and for people that understand that, you can see here that we're working with moderate to high heritabilities for a range of traits there. I've just pulled out a few, and the summary of that is there's large differences between the sires, both in Brahmans and composite, for their steer sons' performance. So no real surprises there. We already knew that, but the key here is, but what about the daughters? What are we seeing here? And for that reason, we wanted to look specifically at female reproduction. What degree of genetic control? Are there new measures we can be using? Are there early in life traits that we can be looking at? What are the relationships to those steer traits? And to do that, we needed records from heifers through to lifetime and retaining those cows. And so that made it a very unique design and a very long-term experiment. So in terms of our cow recording, we had more than 1,000 cows of each of the 2 genotypes. Each cow has full reproductive history up to that 6 breeding seasons. For each cow, we know all the information about puberty, her post-calving recycling, complete mating details, bull in, bull out dates, the syndicate of bulls, etc. Complete calving details, date of birth on all calves, birth weight, DNA parentage, calf survival codes or death codes. For each of the cows, we have weight, fat, condition score every 4 to 6 weeks throughout their life, so we can monitor and track how these cows are changing in their body composition as they progress through lactation and reproduction cycles. We also have eye muscle area, rib, and hip height 4 times a year. So you can see we've got a massive amount of information, not only on the reproduction of these cows, but also how these cows were changing through that annual cycle. We have many other measures, complete disposal codes on these cows, temperament, adaptive scores, teat and udders, etc. But I won't have time tonight to go into that because another part of this is longevity and also cow and calf survival, which is wrapped up in this area as well. The real important experimental procedure that we used was the ovarian scanning, and this was pivotal to the whole experiment, that we could track what that cow was doing in terms of her ovarian activity. And what we're looking for specifically on the surface of the ovary is this structure called the corpus luteum, which shows us that that heifer has ovulated, or that cow. When we first saw that, we used that as our measure of age at puberty, but also as these cows progress through their annual cycles, we used it to measure the resumption of cycling. So for each cow, we're able to see when did she start cycling after giving birth to her calf. And in total, on the database, we have over 356,000 records on ovarian scanning and more than 60 events per cow. So a massive amount of data to allow us to track the reproduction of these cows. In terms of the reproductive traits, obviously that scanning and calving and joining history allows us to look at a lot of traits in terms of puberty, age at puberty, weight at puberty. Was the heifer pubertal prior to the start of mating? We also did a little bit of reproductive tract scoring. In terms of the cow reproduction, we're able to look at, because we're doing this at an early age, we're able to see whether the cow has conceived, whether she's held that pregnancy at the usual weaning time, sort of April, May each year, whether a calf was born, the days to calving measure, a calf weaned, and we're able to construct traits to tell us the lactation anoestrous interval. That is, how long from calving or from when we started to scan these cows until we observed that she'd recycled or started to cycle again. And that's a very powerful measure, particularly in these Bos indicus genotypes where lactation anoestrous has been known to be a phenomenon, particularly in Brahman cattle. We're able to see whether this cow has actually ovulated prior to weaning, and we start to construct traits that looked at breeding season 1 and 2 outcomes, the maiden heifer and the rebreeding first calf cow. And because we kept these cows then for 6 years, we're able to look at lifetime annual weaning rate and lifetime annual calving rate. So a lot of traits, obviously we can only just look at a few of those tonight. But certainly all this information is also available that people can get hold of. We do see, we might hold it there, Andrew, and just get any quick questions that people might have regarding the design.
Okay, thanks David. We don't actually have any questions in front of us at the moment, unless if everybody does have a question about the design and how this project was set up. Obviously it was a fairly comprehensive project and has collated a whole lot of data. So if anyone's got any questions, I'd encourage them to type it in now. By way of background, we do have tonight online about 125 people spread across the breadth of the country, so obviously they managed to drag themselves away from the opening of Big Brother and join you tonight, Jono. So we don't have any questions, so I suggest we'll keep going and save some time for some questions on the results.
Okay, very good. What I want to go now through is just a real snapshot of the results. Yeah, there's lots to give, but for people who aren't familiar with this project, I just want to give some of the big hits and then stop for questions again if anyone's got any, and then finish up with, you know, where do we see this going and what can people start to do. So in terms of the results, the first thing is if we look at age at puberty, I just want to go through a few examples here where we can show that there's lots of variation out there for these reproduction traits. And this first example is age at first CL, which is the age that we saw that first corpus luteum, which would be our measure of age at puberty. So across the bottom of the axis there is just age in days, and then it just gives the frequency or number of heifers. So you can see we get a spike of heifers that are showing puberty at about 520 days or 16 months, and then another group here that we're showing it at 25 months, showing the clear influence that we have variation due to underlying genetics, as we'll see in a minute, but also the big impact of the production system and the environment, here showing the effect of the dry season, wet season, cyclical nature, where, you know, if heifers failing to get pubertal in their first post-weaning wet season, you know, there's a big peak of heifers that had to wait an additional 12 months. So, you know, the environment certainly is influencing it, but what we're able to do in this experiment is then say, well, how much of that variation is actually genetic? And that's what this slide gives us, the heritability, which is on a percentage scale, and here are the estimates of the genetic control of age at puberty in our Brahmans and composites. And to give that a bit of perspective, something like hip height is about 60% heritable, a highly heritable trait. Growth and carcass traits are in that 30 to 40%. So you can see the first key result here is that heifer age at puberty in these two genotypes was moderate to highly heritable, and therefore is under considerable genetic control. And as a result, we see big differences between the sires, between our 50 Brahman sires and our 50 tropical composite sires in the age at puberty of their daughters. And as a quick example here, if we turn that into a CRC EBV for age at puberty, we can see in the tropical composites that there's Mount Eugene 920106 at -3 months and Mount Eugene 94025 on the other end at plus 4.4 months. So that, for people who understand EBVs, you can see that there's a 7-month difference there, and what that relates to or equates to is a 3.7-month difference in the daughter's age at puberty between these bulls. So simply half that difference. So that's a very big effect of, you know, just in the sample of bulls we've used, immediately we see genetic differences. I've chosen tonight not to go through breed after each of the breeds, but just to give you an idea, in the Brahmans there was a -6.1 to plus 5.1 EBV spread in the 50 bulls there. So an even bigger difference between the bulls in their age at puberty of their daughters. If we look at the heritabilities for the early reproduction traits, I've just pulled out a few here again for Brahmans. Pregnancy at mating 1 and weaning at mating 1 in the light blue. So we see now heritabilities in that sort of 10 to 15% range. Pregnancy 2, weaning 2, days to calving 2, and a trait called wet and pregnant at 2. You can see we're working in sort of 10 to 20% heritabilities. So certainly we're seeing, as we expected, lower heritabilities for these 0-1 traits. So when it's a yes-no trait, did you get pregnant or not? Obviously, there's a lot more environmental influences, and just the nature of the trait means, it's much harder to get higher heritabilities, and we commonly see heritabilities for reproductive traits less than 5%. So in these data, clearly we're picking up quite a bit of variation. We see more variation at this second breeding season, and in general the tropical composites, particularly for mating 1, had much lower heritability estimates compared to Brahmans. But even saying that, we do see considerable genetic variation. So whilst the heritability is low, the amount of variation there is still considerable, and there's opportunity to make improvements through selection. This is now a graph of the days to cycle in the first lactating cow in the Brahman, and this is a very key result where we see when we weaned at about the 120 days that we see a group of about 52% of the cows were cycling. So we saw a corpus luteum prior to the weaning event of their calf. We see another group that cycled soon after we weaned the calf, and then a group that had extended lactation anoestrous and didn't cycle until the following wet season. So what this represents in our data is clear evidence that we were seeing lactation anoestrous in our Brahman first calf cows, as half of them weren't cycling until we actually weaned the calf. And obviously that has a very large effect on the pregnancy rate in those first lactation females. So what we're able to do is then say, well, how much of this is genetic? And in both the Brahmans and the composites, you can see here that we get almost a 50% heritability for lactation anoestrous interval in the Brahmans, but also considerable, almost 30% in the tropical composites. So a clear basis for genetics being involved in lactation anoestrous, which has often not been considered in the past when people have been trying to look at this phenomenon. So a very, very clear outcome from this result, from this project, is the genetic basis of the lactation anoestrous. And obviously then we see big differences between the sires in their lactation anoestrous interval. And again, here's an example of lactation anoestrous EBVs from the CRC for Brahmans, where we see shorter lactation anoestrous intervals in the Lancefield 4999 bull, minus 3.3 months, through to the Belmont bull on the bottom of the list, Belmont 7996. He has an EBV of plus 5.6 months. So this represents a 4.4 month difference in the first calf daughters to resume cycling in this experiment. So a very big difference in how long it took the daughters to recycle, and certainly reflecting that sizable heritability. Likewise, in the composites, their spread in EBVs was minus 1.2 to plus 1.2 months. So not as big as the Brahman spread, but still quite a considerable difference between the bulls in terms of their time to recycle, or the length of their lactation anoestrous interval. Then we move on now. I realise I'm giving lots of results, but hopefully people are keeping up. We can estimate the lifetime calving and weaning rates. So we kept these cows for 6 mating opportunities, and over the whole experiment, we can see that the Brahmans, on average, and these are results for Belmont Research Station, where we can directly compare these two genotypes. We can see that the green bars are their annual calving rate. So we across the experiment, we had about a 75% weaning rate in the Brahmans and about 85 in the tropical composites, and the weaning rates are about 65 in Brahmans and closer to 80 in the tropical composites. So a lot of people would say this is very typical of the type of weaning rates, or the weaning rates we see in Northern Australia, and obviously the opportunity to improve them. The other thing to take from this slide is the difference between the calving rate and the weaning rate, which represents calf losses. And this is also a significant phenomenon in Northern Australian production systems, and certainly part of this research, but also a larger project that's finishing up with MLA on cash cow, looking at where these reproductive losses are occurring, in particular some of these calf losses. Won't have time tonight, but certainly this slide bears out that there are significant calf losses also occurring. So if we look at the Brahman results there, we now want to say, well, how much of this genetic do we see as genetic? And we once again estimate the heritability, how much of the differences we see are due to sire and dam differences. And for something like lifetime annual weaning rate, in Brahmans we're getting an 11% heritability, in tropical composites a 7% heritability. So once again, lower heritability for these 0-1 traits, but still considerable genetic variation that we can work with. And obviously, as we go forward, what I'm going to tell you is how much of this genetic variation here is explained by some of those more highly heritable traits, like the age at puberty and the lactation and anoestrous. But in terms of the lifetime weaning rate, although there's a low heritability, because of the large amount of variation we have, here's the spread in the EBVs between the top and bottom Brahman sires. So McKellar Ricardo at plus 15%, the Belmont bull down the bottom at minus 11, so representing a 13% difference in daughters' annual weaning rate between the top and bottom bulls of the snapshot of 50 bulls that we used in this study. So clearly a large variation and a real opportunity to improve these traits through selection. Just quickly, in terms of the composites, once again, lower heritability, but still an 11% EBV spread in lifetime annual weaning rate. So this is each year the daughters of those bulls are calving at those increased rates. So quick summary thus far. Genetic control of these reproductive traits is clearly apparent. The component traits like age at puberty and lactation and anoestrous are more heritable than the 0-1 yes-no type traits, and we see large differences, large genetic differences at that first rebreed. We see more genetic variation in the Brahmans for a lot of these traits, and these lactation and anoestrous traits, whether it be the interval or the days to calving measure, or was she cycling before weaning, we see lots of genetic variation, and obviously that's key to the next set of results. So just one more slide. I'm just going to try and summarize it in one slide. What are the key female trait relationships? So we measured lots of traits, and what's it telling us? The repro measures, so whether it's conception rate, pregnancy rate, days to calving, weaning rate, calving rate, they're all very similar genetic measures. So we can use one and interchange it with another. And most importantly, the days to calving, which is the trait that's used in breed plan, it has a very high relationship genetically with age at puberty, lactation and estrus interval, and the lifetime traits, which is pivotal in understanding how we can go forward. Those people who've been using days to calving EBVs have been capturing the genetic variation that existed in age at puberty, the genetic variation in age at lactation and estrus interval, and the genetic variation in lifetime. So that's a very nice outcome from this project, that trait that is in breed plan is capturing a lot of the variation that we saw in this study. Age at puberty is moderately useful as a measure to capture as a measure of lifetime reproductive performance. It mainly captures the genetic variation in Brahmans for the first mating outcome, but is also for tropical composites a good predictor of that lactation and estrus. So age at puberty, whilst we could use it, isn't a single measure that could replace any of the other measures that we took in the study. However, first lactation and estrus interval is a very useful measure of lifetime reproduction. A lot of the variation that occurs in lactation and estrus explains the variation that we see in lifetime reproductive performance. I haven't told you much about it, but we measured that body composition on these cows as a single predictor of reproduction. It was only limitedly useful, so limited usefulness. So measures of fatness, measures of eye muscle area, measures of body condition score, whilst it does have relationships with reproduction, those genetic relationships are not very strong, and so not a useful single or replacement measure for the for measuring reproduction. I might take questions. I'll just finish one more. Can we select for steer traits and cow traits? What has this experiment told us? So for every daughter that a bull leaves out there, for every steer he produces, he's also leaving a daughter. So can we select for improved carcass without compromising reproduction? Because we've now established that reproduction is clearly has a genetic basis. In summary, the experiment would tell us yes, we can do both. There are no really strong antagonisms between, so genetic antagonisms between doing both. So we can have both, but obviously we need to measure them if we want to improve both. So if we want to find cows that are good for reproduction, or bulls that have got good daughter reproduction and also good carcass, we'll have to measure. We can't simply rely on measuring one and getting the other. So whilst it's good on one hand we have no antagonisms, it also means that we don't have the opportunity to measure both traits. Of course, if we don't measure the traits, then by chance you can end up with unfavourable combinations. So if you simply select for carcass traits and don't measure reproduction, then there's plenty of chance that you will end up with reduced reproduction and vice versa. And the other point is that the Brahmans are clearly quite different between the genes that are involved in a feedlot finishing steer compared to a lactating cow, and that probably doesn't surprise a lot of people, that a steer standing in a feedlot putting down fat, the genes for fatness in the cow under lactation are obviously quite different, and we see quite a bit of re-ranking between the sires if we look at fatness of the steer versus the fatness of a cow going into mating as a first calf cow. So Andrew, we'll pull it up there if there's any questions, because that's a really quick 20-minute run through of the key results from the project. Obviously there's a lot more, but in terms of capturing what we saw for the cows and in terms of female reproduction, they're the key results. If we take some questions, then I'll come back and say, how do we capture that? Particularly, people that are Angus breeders, people might be not in these genotypes. How do we go ahead of capturing those traits?
Okay, thank you, David. We do have quite a few questions that have come in, so I apologise to people if I don't get to your question, but we'll hope to get through as many as we can. Just one point of clarification to start, David. What percentage of Brahman heifers were cycling within 1 month of having their first calf?
I don't have that number off the top of my head, but people have to understand we started to scan these cows at the end of calving. Because we have cows with young calves and also cows that are still pregnant, we couldn't bring these cows into the yard to scan them. So scanning started at the end of the calving period, and it was also the start of the joining period. So on average the cows were sort of 4 were 6 to 7 weeks post-calving when we started to scan. And so some cows, we picked up a CL on that day of the start of mating, but we didn't actually know when she calved. That's why I call the trait lactation anoestrous interval without confusing people, but it's not strictly what people might know as postpartum lactation anoestrous interval, or PPAI. So, I would have to go and look at that graph again of how many cows were straight after calving, but certainly there were some cows from what we can see that did, even in Brahmans, did cycle within a month of calving.
Okay, and next one here is, did the Brahmans display the same age at puberty profile as the tropical composites? That is, a peak at 16 months and a second peak at approximately 26 months.
Yeah, most definitely. And in fact, because we also had them at Swans Lagoon, we also got another peak, another 12 months later than that, for some really late heifers. You can also do plot weight at puberty, and then you tend to get a more bell-shaped curve. So whilst there was a big effect on age, what you tended to see was a bell-shaped curve, and for Brahmans that was like 340 kilos, but going down to as low as sort of 250 and up to 450 kilos. So we see that variation in the Brahmans and the composite, both for age and the weight, but certainly the age was affected by that wet and dry season.
Okay, and the next one, is it possible there's less spread in the time between cycling for tropical composites than Brahmans due to more emphasis on this being placed on this trait in the development of the breed?
Yeah, that is certainly possible with the way they were bred and developed at the in terms of the Belmont Red at Brahman, at the Belmont Reds at Belmont Research Station. There is a little bit that they were in more favourable environments, so we didn't have the tropical composites at Swans Lagoon, but we still saw the difference at Belmont where they were treated alike. So whilst we do see that the environment will cause extended lactation anoestrous, when they were treated alike, we did see that the composites still had that advantage. Some of it could also be retained hybrid vigour in those composites because of the way they've been bred, and certainly that could be affecting and helping their reproduction as well, because we know crossbreeding is another way to increase female reproduction through hybrid vigour.
Okay, and just one last one before we get back underway, and this just comes back to a question I missed before on the design. In your design, was there a crossbreeding type of design versus just straight purebred, I suppose, if we're looking at tropical composite and Brahmans being purebreds in that sense?
Yeah, in terms of the design, no, we were treating these as two breeds. I called them genotype, but the Brahman breed, and then we lumped all the composites together and called them a composite breed. Now, the composites were from NAPCO, from AA Company, and also the Belmont Reds, and so in some cases they are a crossbred genotype. Even though we've called them a composite, in some cases they were more F1 type genetics. So we do know that we've got hybrid vigour in those composites, but in our statistical analysis we're looking to control that. So the only difference was at Belmont Research Station we did join them in Brahman and composite joint joining groups, so we didn't break the two up for joining, so they were directly comparable for their whole lives. In that case, we did use Brahman sires and tropical composite sires and generate the reciprocal crosses. So there was some of that at Belmont, but that was mainly just to allow us to get good head-to-head comparisons of the reproductive performance of those females without breaking them up for even for joining.
Okay, thank you for that clarification. So there's a few other questions in, but just because of time and obviously with a reasonably big audience tonight, we might get back underway, David.
and try and some of those questions we haven't answered we may have time for at the end.
Yeah, well, if anyone does have a question, I've been pretty good on the time, so now I just want to go through how we might capture it. So it looks like we will have a little bit of time at the end, so if anyone's got a burning question, rip it into Andrew and we might have time to cover it. So what I want to go now is, how do we go about capturing some of these genetic differences in female reproduction? So what have we got to do to harness the sort of differences we've seen in the CRC, recognising that we've just a snapshot of what's out there and what would be possible. And I like to say, how do we get more of these sort of cows? Now, if you look at those two cows, they're both from the experiment. One's a tropical composite and the other's obviously a Brahman. Now, there's nothing particularly surprising about those two cows as they stand in front, and that in itself is pretty good example of exactly what you can't do. You can't really see reproductive performance. But in this case, I can tell you that they're both 12-year-old cows. They're both still alive, and they are both now pregnant for their 10th time from their 10th mating, and have weaned 9 other calves right through to weaning. So to me, in our production systems, whether it's north or south, that's the type of genetics that we'd like to be able to identify. And I've just pulled this out from the Brahmans in the experiment. I've got a group of cows on the left and a group of cows on the right, and I could have put hundreds of cows in these slides, but I've just pulled these out for examples. So these are some of the cows that have gone through the experiment, and you can see they're all shapes, sizes, colours, body composition, hip heights, but there's a clear difference between those two groups of cows, and it's very challenging to look at that, to know that these cows on the left have weaned 3 calves from 6 opportunities, and these cows have weaned 6 out of 6. They haven't missed, and they're across Swans Lagoon, Toorak, Belmont, so across all the different environments. We see some cows have hit 6 out of 6, even at Swans Lagoon, which is a pretty tough environment for people who don't know Queensland, coastal, air, up near Townsville. And then we've got the cows under exactly the same conditions, only managed to wean 3 out of 6. And in many cases, those cows that weaned 3 out of 6 matings exhibited the classic hit-miss, where they failed to calve every second year. And so my question is, how would we know that difference if this is in any breed? How do we actually capture it? And there's one answer, and that is to record reproduction. If you've got the reproduction records, then you've got the power to start to do something about it, particularly as we see the heritabilities that have been exhibited in this project. So how do we go about recording it is what I want to now focus on. And what we're going to do is build this all into an EBV. So people that are familiar with breed plan would know that we have reproduction EBVs. What we're now moving towards is, how do we capture that age at puberty from that maiden mating, given that it's more highly heritable? We then want to capture that lactation anoestrous in that first rebreed and then subsequently. And we also now have the opportunity to capture additional traits. So we currently use scrotal size in our female reproduction EBVs, but we have now the opportunity to look at more semen and sperm traits, which is the topic of next Monday's webinar if people are interested in that. And also Mike Goddard will be talking about DNA genomic predictions that have come out of this work. So the plan is to start to harness all 4 sources of variation into an improved reproductive EBV for Northern Australia, but not exclusively for Northern Australia, because we have the opportunity to also expand this into the South, and that we're able then to describe those differences. So the first job of any EBV is to describe the differences that exist in the population, and then the second is to use selection to improve it. And the power of selection is to use the superior animals to drive the average of the population or the mean of the population forward. And in this case, we have to be looking at replacement bulls. Simply culling cows, as I'll mention in a moment, won't be driving us forward. We've got to be identifying the superior genetics in our young replacement bulls. So for bull breeders, these are sort of like the take-home messages. And this is for any breed. Obviously, this research has been in 2 tropical breeds, but certainly Santas, Drought Masters, Brangus, these results are directly applicable to you, but I also believe that temperate breeds in southern Australia can learn from these results as well. And so for bull breeders, what we're seeing is that we've got to record that female reproductive performance. But the focus of the recording should be on the maidens and those first calf cows. If we capture that information, we're capturing the most heritable part of the female reproduction. We get those records earlier and we can make selection decisions. So we need joining details, we need calving and weaning outcomes, we need days to calving type records, and I also think there is the opportunity to use ovarian scanning, particularly for something like age at puberty, but that would be possible. The lactation anoestrous, scanning lactating cows, is a bit more of a challenge from a management point of view. But certainly we've got to capture that female reproduction data. Then we've got to apply maximum selection pressure to those maidens and first calf cows. We've got to really cull heavily and remove those genetics from those seed stock herds on those maidens and first calvers. We've got to tighten, there's the opportunity to tighten joining periods for those maiden heifers. So if we can do 6-week to 9-week joinings on those maiden heifers, that will really put the pressure on the age at puberty component and make sure that we're getting the best genetics in those heifers to start. Obviously, we've got to then include reproduction in overall selection. As we see, the steer traits are highly heritable, economically important. We've now got to put the reproduction and those other traits together when we're making bull selection decisions. And as I said before, the genetic progress, particularly for reproduction, will come through improved sires. That's where we've got to drive it forward, is using better bulls and making selection decisions on female reproduction traits. And as I said, Mike's going to talk about the DNA tests and the new bull measures, which we can also work into here. I'll talk about next week. So that's the bull breeder. What can the bull breeder start to do to capture those genetic differences in any breed? Commercial producers, how can they use the results? Well, the first one is buy bulls with above breed average reproductive EBVs if they exist. So if breeds have days to calving, if breeds are doing scrotal, then commercial producers need to be seeking out those bulls that are above average in their breed. And that's where the improvement will come from in those commercial herds, is buying better bulls. Buying better bulls that have better age at puberty and less lactation anoestrous is a real key. If a breed doesn't have reproductive EBVs, and you can get on the web and look through the calving history, my best bet at this stage is to avoid a dam. So avoid buying a bull if his dam has missed as a first calver. So if she hasn't calved and then he's the subsequent son, then if the heritabilities of these traits would suggest that that would be one way that you could pick up a bull that might be above average for reproduction. So buying bulls is what we need to be doing. But then on the female side, again, restrict the heifer joining and, if possible, cull non-pregnant heifers. So mate, almost overmate heifers to keep as many heifers as you can, but then cull very much on those that are non-pregnant. Also, if possible, cull non-calving cows, because we do know that they will be passing those genes on to the next generation. And often we see it's the cow that takes the year off that's the one that produces the replacement heifer, and worse, the replacement bull. So there is some things you can do on the culling side that will help the genetics of the commercial herd. However, if it's not possible to cull cows because just to maintaining herd size, obviously we have tough years, but also if you take a big hit on those first calf heifers, if it's not possible, then one thing we should try and do is avoid keeping future replacements from a cow who did skip as a first or second calving cow. So if they do skip and you retain them, then we should be trying to see if there's a way of avoiding keeping a replacement out of that cow in the future, because that would be trying to, at least at the commercial level, stop the multiplication up of below average type genetics. So there are a couple of take-home messages. Hopefully they're useful to people. As I said, more next week about how we're going to build in the male reproduction and what that has to offer from measurement perspective in terms of this whole female reproduction. But certainly the opportunity there to lift weaning rates in Northern Australia, and particularly from these results, through selection for improved reproductive performance. So a real opportunity there to use better genetics to increase those weaning rates. Obviously I've flown through this fairly quickly. Opportunity now for some questions, but feel free to scribble down that email address if anyone has any further questions or any follow-up that they'd like to do. And just in finishing, Andrew, just like to put up an acknowledgement slide. Obviously this Beef CRC project and subsequent work has been a big collaborative effort, obviously through the CRC and its core partners, but also a lot of cooperation, a lot of funding, MLA and ACR, our fantastic cooperators who continue to maintain ownership of these cows right through the experiment, and also a great team of technicians and scientists and scanners and everything. And of course the whole project wouldn't have been possible without Queensland research stations that ran these cattle. We had impeccable bull control, and if you're going to do any reproduction work, you've got to have good bull control, and it was second to none given the management and the staff that we had on these research stations. So I'll pull it up there and take any questions. Thank you.
Okay, thank you, David. We do have quite a number of questions in. Not quite sure where to start, actually, looking at the list. The first couple just relate to the feasibility, I guess, of scanning scanning ovaries. First of all, I guess the first question is, how often would they need to do it, and what's the availability of scanners? And the second part of that, coming from another person, is, given some of the difficulties in scanning for ovarian activity, how much increase in genetic progress would we expect as a result of scanning ovaries, and is that cost effective?
I think we'll have to continue to do some sums on it, but I think there's an opportunity, particularly for if we have breeds where age at puberty is an issue, then a once-off scan we know will produce a heritable trait that we could use in selection. So, but we also have the possibility of doing a repeat scan if that was available to even get a better measure. So I think really age at puberty, yes, we could see that there could be clearly an advantage of doing scanning if age at puberty was a problem. There are scanners out there, but now that the CRC is finished, they're going to be harder to get, but we can always train new people.
Okay, thank you. And I guess if anybody has any questions about accessing scanners and things, then if they want to contact their SPTS or TBTS technical officer, we may be able to assist you with that a little bit more. The next question is, do we risk ending up with smaller cattle if we focus on earlier maturing bulls only?
If you're looking at selecting for maturity pattern, then we may change the size of these, but certainly all our work we're looking at, we can estimate the genetic relationship with weight, with hip height, and everything, all the evidence in both the composites and the Brahmans is that we can select for reproduction without making our cattle smaller. In fact, we can make them bigger, we can make them smaller, we can make them fatter, we can make them leaner. So there aren't any real strong antagonisms between physical size and reproduction. So that would be how I would look at it, is we can select for reproduction and also multiple traits select for whatever we think is an appropriate size of those cattle.
Okay, the next one just relates, I suppose, trying to relate this to the south. In southern beef systems, they often involve shorter joining periods, heifers calving at 2 years of age, and strict culling for fertility. Are southern producers already selecting for many of the reproduction traits that you have identified?
Yes and no. In southern Australia, if short joinings will be putting the pressure on, but I would argue that we're only culling. We're not actually actively selecting. So what we want to be doing even in the south is pouring better bulls in there. If we're only ever culling, we're not actually making a great deal of genetic progress because we don't have a lot of selection intensity in our cows to start with. So whilst culling is okay, we're not really identifying the genetically superior bulls who we want to be the size of the next generation. So there are things that we can do in the south that will be helping, but we're not going to make a lot of progress if we're only just culling those non-calvers.
Okay, this one relates to, can using CEDA programs to tighten calving in maiden heifers cause an issue for results in calf rates for these heifers?
Yeah, so the use of artificial breeding technologies, currently when we do our analyses and breed plan, we don't use that data. For that exact reason is we don't know what is the real effect. So we've got work going on at the moment to collect better data to see if we can use AI data, and particularly AI data from synchronization programs, but at this stage we don't use that data because we don't know whether we are actually selecting for inherent reproductive or fertility, or if we're just selecting for those that respond to synchronization. So yeah, difficult to use that data at the moment, but we're looking into hopefully using it in the future.
Okay, we'll just have one or two more before we pull it up. Basically, you referred to a number of EBVs that you'd calculated for the different traits. Typically, what were the kind of accuracy of those EBVs?
For those highly heritable traits like postpartum anoestrous interval, the accuracy on those EBVs for those bulls will be 80% plus. For the lowly heritable traits, we're getting accuracies probably 60 to 70% for our bulls. So just depending on the heritability, but because most bulls had 25 daughters, we got very accurate EBVs for a lot of these traits.
Okay, well, there's a few more questions, and I apologise to those people that we haven't got to tonight. I've endeavoured to get to as many as we can, but just for time purposes, we might pull it up there, and very shortly I'll put up a list of people who you can contact in addition to David's contact details, which he very generously gave us to follow up on questions after this webinar. So David, if I can just get you to change the presenter back to myself, we'll just go through a very small amount of just housekeeping to finish up tonight's webinar. And again, we'll just have a slight delay while we organise our technology.
So are you getting that, Andrew?
No, I'm not actually, David.
Make presenter.
Do you have to select, you might have to select me when you, that's better, yes.
Good.
Okay, so just before we finish, just a little bit of housekeeping. We just have 2 questions, and I apologise for surveying you at the end when you're all probably ready to go. But we'd just be interested in your feedback from tonight's webinar. So I'll very quickly just put up a poll which should come up on your screen. What we get you to do is just whether you believe that the webinar tonight was useful. If we can just get you to go through and click between strongly agree down to strongly disagree and the different variations in between, just a way of us collating some feedback on tonight's webinar. So we'll just leave that open for a little bit of time while people vote. Okay, so we'll close that one off now, and just one last question for you is basically based on this webinar. Do you feel that you're more informed now to actually about the application of the outcomes from the beef CRC within your enterprise? And again, if I can just get you to click between the strongly agree down to the strongly disagree. Okay, so I'll close that one up now, and thank you very much for your participation in that. Just some other housekeeping, which we also, sorry, I didn't press the correct button. I'll close that one off now. So some other housekeeping which we've got is obviously there's a lot of information which David's put up in front of us tonight, all very valuable information. For those of you that would like to go and just view certain parts of it again, there will be copies of this webinar made available from the SPTS and TBTS websites, and also you will be able to download a copy from YouTube. So if you go onto the YouTube website and sort on SPTS or the cutting edge or that type of thing, you should be able to bring up the video of tonight's and watch tonight's presentation again. For those of you that may have had difficulty with some of the technology tonight and have problems downloading it over the internet, we do have a CD available which will be made available at the end of the webinar course, so in 6 weeks' time. If you just ring up your relevant SPTS or TBTS technical officer, we'll put you on the list and that will be sent out to you free of charge. Also, there will have, for those of you that were interested in attending upcoming webinars, so this course is running over the next 6 weeks, and obviously David will be back with us next week presenting some of the information that he found from the male counterparts as part of this project. You will need to register separately, so at the moment you do need to register for each session that you're interested in. For those that have further questions, obviously we didn't get through all of those tonight. You do, however, put up the information in front of you there for the relevant SPTS or TBTS technical officers in each breed. So you'll have their contact details hopefully at home. But if you give any of these people a call or the person relevant to your breed, then we should hopefully be able to provide an answer to any of the questions which didn't get answered tonight. And if we can't answer them, we'll be able to get in touch with David and provide you with an answer. So before I close, it would be remiss of me not to thank David very much for his time tonight. As usual, he's done an excellent job and presented quite a complex topic in a very, I think, to me, very simple and comprehensive manner. So on behalf of all the attendees and particularly the SPTS and TBTS team, I'd like to thank David very much for his involvement and also thank you all for coming in and attending. And with that, we might end tonight's webinar and hopefully see you next week.